Well, I'm really happy to be part of your Institute celebration today of World Metrology Day. I am currently working in the chemical and biological metrology branch of NMI Australia. I run a section called Chemical Reference Value. The theme for World Metrology Days this year provides a very timely opportunity to review the contribution of metrology in food testing. My presentation today would cover some of NMI Australia activities in food, especially for chemical metrology. First of all, I would like to start with an introduction of our institute. It is part of Department of Industry, Science and Resources. It was formed in 2004 from the amalgamation of three organizations: Australian Government Analytical Laboratories, National Measurement Laboratory, and National Standard Commission. And in 2010, trade measurement was added to the institute. Each organization brought very different measurement expertise. This is why today NMI Australia is able to deliver a very unique range of activities. Our role is defined under the National Measurement Act, and it states that NMI is responsible to coordinate Australian National Measurement System, to establish, maintain, and realize Australia units and standard of measurements for a range of measurements: physical, chemical, biological, legal, and trade metrology. So I can say that we are the reference point for all measurement activities, which enable production of reliable, traceable, internationally accepted measurements. Just to give you the NMI structure as it is today, NMI is headed by Dr. Bruce Warrington, which is our CEO and Chief Metrologist. In the current structure, we have 5 main branches. First branch I talked about is Physical Metrology branch, which focuses on measurement of a wide variety of physical quantities. And I think they have pretty much conducting similar activities to what your institute does. We have a Legal Metrology branch. We have in the morning a speaker about legal metrology in New Zealand. Our Legal Metrology branch administers trade measurement laws, including measuring instruments such as petrol browser and supermarket scales. It develops legal metrology policies and provides what you call pattern approval for measuring instrument used in law enforcement. One example I can give you is traceability in breast alcohol analyzer measurements. And my section, for instance, is very involved in producing the solution for this type of analyzers. So chemical and biological metrology branch is the area where my work resides. We produce chemical and biological reference material, which are used as calibrants or to validate routine analytical methods. Next branch is Analytical Services branch, and this is a very unique branch to NMI Australia. It performs a wide range of routine analysis in the chemical and microbiological space. And for us, they are a valuable source of information when we want to find out what are the current analytical measurement issues. Last but not least, the Strategy and Operation branch provides a number of ranges to the other 4 branches, such as Business Strategy and Sustainability. It has the Quality, Safety and Risk section, as well as International Relations. Each of the branch brings valuable contribution to food sector. So we conduct many activities related to food in our institute. Mainly to maintain and advance the quality of food metrology. We're doing a lot of research, as well as we provide a scientific leadership role. Today, I selected a number of examples of such activities to cover in my presentation. So let's start with what do we do to maintain and advance quality of food metrology. So our main role as a National Measurement Institute, as you all know, is to provide measurement traceability. For many of physical quantities, these are underpinned by extensive and effective calibration and traceability systems. So establishment of traceability for these quantities is relatively straightforward. For chemical measurements, there are very many challenges. First of all, there are a huge numbers of chemicals, and each one of them requires its traceability path. Second challenge is to establish the traceability in chemical measurement. We need two things. One is to confirm the identity of the chemical as well as the quantity in a matrix. For identity confirmation, we need reference material, pure substance calibrators. And to provide the value of an analyte in a matrix, we need matrix reference material. So we need this different type of reference material to be produced. The production of the materials take quite some time, sometimes from just a few months to up to 2 years, depending on the complexity of the materials that we produce. So one of our roles is to prioritize which one of them we produce, and we do that in careful consultation with a huge number of stakeholders. And to characterize these materials, we need to develop reference methods. So you can see in this pyramid, highlighted in gray, where our contribution is to the link between the SI unit, the abstract SI unit, and the routine laboratory testing. So developing reference methods help us to produce what we call reference value, which are of high accuracy and low uncertainty, and we use them in two areas: to characterize our certified reference materials, but we also to provide a metrological traceable assigned value to our proficiency testing program, which is greatly appreciated by our testing laboratories. And I'll provide some examples as we go now. So first of all, looking at pure substance organic calibrator, we have about 60 pure chemical reference material for agrichemicals, veterinary steroid, growth promotant, antibiotics, and key metabolites. So about 60 materials in the area of food out of about 300 materials in our collection. We are also the curator of Australian Pesticide and Veterinary Medicine Authority, and we take care of about 800 chemicals that if they are needed in emergency, we can provide them. So looking at producing pure substance organic calibrators, we have to, as I said, do two steps. One is to confirm the identity of the analyte of interest. And we are very fortunate in NMI Australia that we have a section that can do synthesis of chemicals. So they synthesize a chemical, and then they have to use a range of spectroscopic techniques to confirm the identity. Some of the spectroscopic techniques they have available is mass spectrometry, infrared spectroscopy, or nuclear magnetic resonance. The second challenge after they confirm the identity is this section has to assign the purity. And being a metrology institute, we have to make sure the purity assessment is done without with the absence of a bias. So what do we do in this space? Usually we have to use two complementary approaches, for instance, mass balance and quantitative nuclear magnetic resonance. This is a primary reference measurement to value assign the purity of organic material. Once we have the identity and the purity, we then sell our physical material. Example here in the photo is zearalenone, accompanied by a certificate of analysis, which it's a very complex certificate and contains a lot of information about the material and how it can be used and stored. So once we have our pure substance reference material, we have to go and produce matrix reference material. And this is pretty much my section. To certify matrix reference material, we use high accuracy method with very low uncertainty and established traceability to SI. To do that, we use primary methods, example, isotope dilution mass spectrometry, which is a primary ratio method. And sometimes in inorganic analysis, we use standard addition. To demonstrate the performance of our method is through participation in international key comparison with peer NMIs, which are organized by International Bureau of Weights and Measure. Let's look at an example of us participating in one of such international key comparison. Bovine liver. We did a couple of trace elements. In the photo is nickel. Our results were excellent. And once we demonstrated our capability for this type of measurement, we can use this capability to characterize trace certified reference material, as well as provide reference value for proficiency testing program. So one of our matrix reference material in our collection is trace elements in prawn called MX009. This material was certified initially in 2010 for one element mercury. And from the feedback of our participants in PT program and from the user of the material, we actually started a program of recertification, which we finished, I think, a year ago. And we added 11 more analytes. We certified for 11 more analytes, which were chosen either because the participants reported they are common tests for this type of matrix, or because there was evidence to suggest they had particular analytical difficulties. This is the certificate for the material. So we have 12 certified value, but also in the certificate on page 6 of the certificate, actually, we produce some information value which we obtained from running some inter-laboratory comparison. So looking at our proficiency testing program, we run about 20 studies per year. The team is very small. We have only 4 people to do these studies. And we do everything in NMI, from designing the PT study to invitation to data analysis and producing the final reports. Almost all our PT programs are accredited to ISO 17043. In the space of food sector, we cover trace elements, polyperfluorinated alkyl substances, pesticide residue, folic acid, and some allergen. This food program is very important for our stakeholder from 2 perspectives. One, it demonstrates the quality of measurements results given by laboratories. And second, it increases the stakeholder confidence to make critical decisions that affect the food supply chain. So let's look at some examples from our PT study and see how we reference value can help participants. So this is an example from our PT study in prawn, where we provided reference values for 2 analyses that were deemed to be some difficult analytes for this matrix, nickel and chromium. Codex Alimentarius Commission recommend for measurements of elemental impurities in food sample, digestion until extraction is complete. What does this mean for laboratories? Is that they are expected to report total elements in food samples. And to achieve total elements in food samples, they have to use an aggressive digestion regime. It could be either a high ratio hydrochloric acid per sample size, a high digestion temperature, or hydrofluoric acid. Now, hydrofluoric acid in recent times are not used often because it has some potential health and safety effects. So laboratory avoids using this type of acid. And what you can see here is that what we highlighted in red are the laboratories that use some of digestion regimes that are a bit more aggressive, and their results were much closer to the reference value than the other laboratories. Another example I want to give you is where PT helps participant laboratories identify issues with their analytical testing. What we did here is we spiked gravimetrically bifenthrin in tomatoes. Our reference value, as you can see, it's in red. It's very close, aligns very well with gravimetric spiked value. And we did spike the matrix to a level that was well below the MRL, which is maximum residue level admissible for tomato, bifenthrin in tomato. There were laboratories that have difficulties with this pesticide. But what I want to highlight here is that laboratories 3, 20, and 2 not only have difficulties with the analyte, but they reported results which were above the maximum residue level, which made the tomatoes not be compliant. And the reality was that the level of bifenthrin was way below the maximum residue levels. So this laboratory would have to investigate issues with their methodology. So I talked to you about two types of materials, pure substance calibrators, as well as matrix reference material, and then about the proficiency testing program. Where do all these materials and PT program fit with the routine laboratory analytical testing? So let's have a quick look. So matrix reference materials, which usually mimic routine sample, should go into the process right at the beginning, starting from subsampling the sample. Our calibration reference material should be used by routine laboratories for instrument calibration. And also, matrix reference material and calibration reference material should be used for when the laboratory are actually validating the methodology. Once the analytical laboratory finish their testing and they get a measurement result, they should take these results through a verification step by using proficiency testing. So as you can see, our materials should be really helpful in a routine laboratory testing process. So now let's look at some of the research activities we do in NMI. Analytical laboratories around the world, including the analytical laboratory in NMI, develop methods for a class of emerging contaminants called poly and perfluorinated alkyl substances, which are a large complex group of about 9,000 of chemicals. For these 9,000 chemicals, only about 45 methods are developed to date. These contaminants are very resistant to degradation and accumulate in food chain because and became of high interest to community due to a contamination incident when using legacy firefighting foam in Australia. Being complex chemical, the infrastructure was needed to ensure quality and comparability measurements. And to help with this, NMI developed the first national polyperfluorinated alkyl substances proficiency testing. It was not an easy process. The field was really new for us as well. And we had to develop a preparation method for fish and prawn. These were the first matrices we did in food. And why was that important is homogeneity of proficiency testing samples are very important. We don't want that the results produced by the variation in results produced by analytical laboratory to be due to inhomogeneity in our samples. And we had some issue because, as you can see in the photo, we had to take a whole fish and homogenize it well to become just a small tube. To do that, we need to develop a grinding method that was able to give us the right homogeneity. And we developed a cryo-grinding method. We tested both fish and prawn using this method, and we were very successful, both matrices now passing the homogeneity test. And I want to show you some results from a proficiency testing where both matrices were introduced, fish and prawn. Participants seem to have a lot more difficulties with the prawn matrix than the fish matrix. Let's look, for example, at PFOS. Fish results were really tight. So there was no much variation between the results, where with prawn, you can see there is a large variation. The horizontal black line is the assigned value, which was done from the consensus of participant results, was red triangle, it's our spike value. And for all three analytes, the consensus from participant laboratories was much lower in prawn than the spiked value. So it is clear to us that participants had low bias issue for prawn samples. And this highlighted the fact that you can't really develop one method to for all matrices. You actually need to participate in proficiency testing and see how your results fare against other participating laboratories, and maybe you need to tweak the methodology for different type of matrices. Going now into a different area, let's look at our bioanalysis group. They developed a method for identification of animal species and quantification as relative proportion using a technique called digital polymerase chain reaction technology, what we call digital PCR. They developed this area for a variety of reasons. It's to ensure that buyers get what they paid for, to check for meat adulteration, to prevent spread of disease, and of course for food safety and correct labeling. NMI is the only laboratory in Australia that developed relative proportion capability for a variety of meats. Currently, they are developing this method for camel, donkey, and crocodile. And I was very interested to find out why this particular matrices. So I asked the analyst, and they told me, for instance, for camel, they looking to develop the method because recently there was an incident with pet food where camel meat was used, and the dog died by consuming that food, and that's because camel has a protein that is not really good for dogs. And the next area that I like to have present to you is that nanometrology area, and we're looking at distribution of particle in food. Nanoparticles are particles with at least one dimension in the range of 1 to 100 nanometers and may occur either naturally or being added to food, such as enhancement or antimicrobial, or leach from packaging, or purely being just a contaminant. This material may have different properties from the same substance when it's in bulk. And because of this different property, it may lead to different interaction with humans and with the environment. And for this reason, nanomaterials are under scrutiny at the moment. Why does it matter? It matters because, for instance, in EU, they mandated labeling of products containing substances in nanoform. In Australia, currently, there are no provisions specifically referring to nanoparticles, but we should be ready just in case there is a change. And because there are some safety concerns of nanoparticles biodistribution in human, for instance, when consuming food. There are also currently a number of challenges with measurements of nanoparticle in food. The measurements are very difficult, and there are limited available standard methods and reference materials to help with method validation. So our institute is involved currently in a collaborative project regarding titanium dioxide particles. So this collaborative project is between NMI, ANSTO, and CSIRO, and looking at transport and bioaccumulation of radio-labeled titanium dioxide particles through a rat model using gamma tracing. NMI initially assessed the dispersibility of the sample by sonicating them in different media with different pHs to represent different compartments of the rat intestine. And their conclusion at the time was that the particles remain highly aggregated, which this seemed to be posing a problem. The first phase of the project involved force feeding the rat through a tube. And After 7 days, a tracer was found in a number of organs, but also unexpectedly in lymph nodes, even though the tracer was not found at all in the blood. They thought this problem came from the way the rat was fed, force-fed. There were other aspects as well, and they decided the second step, which just finished recently, is to be repeated using this time solid food. And in the experiments, they use a solid food Nutella. There were better outcomes with the delivery model when using solid food, but further work is required to establish the take-up in the organ versus what passes through the digestive system. And the big picture, the post-mortem PET scan, shows you the tracers, which are kind of blue-greens, the distribution in the red. Last but not least, I would like to present some case study from my colleagues in analytical services branch. Analytical branch is helping with protecting the Australia international trade by offering some testing for compliance monitoring and investigative analysis when using regulated veterinary residues and agrichemicals in exported meat and dairy. They provide rapid response capability by developing analytical methods for low-level contaminants. Another area analytical testing is very well invested is supporting indigenous bush food producer. We assist indigenous business to get their produce to market, and we do this through developing method of analysis, advice, as well as providing a platform for broader awareness through NMI-led events and forums. We have a PhD student that works with NMI, and part of his work at the moment is to assess the dietary safety of Australian native plants, for instance. Australia has a high number of population with various allergy. So analytical testing branch has also a lot of work in food allergen space. We develop expertise in testing food to detect presence of allergenic contaminants, covering a range of allergenic and intolerance-related proteins. At the moment, we have 23 type of tests, developed tests such as for gluten, peanut, egg, milk, seafood, and sesame. So after research, I'd like to show you some of the areas where we took a scientific leadership role. For instance, organizing major events in food sector. Just to let you know that some of these events are coming soon. For instance, the Food Allergen Management Symposium with the Allergen Testing Special Interest Group is going to be held very soon in Brisbane. We partner with Australian Allergen Bureau for the organization of these events, and we have expect participants from international food allergen management community. Some of our major players being clinician, tech supplier, testers, accreditation bodies, legislators, and consumer groups. Another event which will come in November is International Conference of Food Analysis, Melbourne, and NMI Australia is really a major organizer of this event. This year, team aligns very well with World Metrology Day team. The team is Global Food Sustainability in a Changing Climate. And last is Collaborative Research Symposium, where we allow researchers to present their work to a wider audience. Future work for us in food space, these are some of the projects I chose to present to you. In allergen and microbial hazard, we're looking at novel food sources, novel proteins, as well as native grain. Nanometrology, currently we are developing a method for single particle analysis, isotope coupled plasma mass spectrometry, using isotope coupled plasma mass spectrometry in order to produce reference material in food. And in chemical metrology, we are about to release a new certified reference material for pesticide residue in tomato. And we are assessing currently the strategic need of Australian stakeholders for various certified reference material. This is the end of my presentation. I'd like to acknowledge Dr. Annette Koo and Dr. Blake Hall. Thank you for your kind invitation. My NMI colleagues that contributed to this presentation and participant laboratories in NMI PT study whose results are used here. Thank you.